give which of these does not provide three-dimensional images. So we have here atomic force microscopy, confocal scanning laser microscopy, and differential interference contrast microscopy and phase contrast microscopy. So, so these are not usual whatever compound microscope or anything, but these are specialized microscope, each one of them. So each one has their specific function, which we will see one by one. So first we'll see the atomic force microscope. So what is different in this? That we have a cantilever and we have a tip, and laser is also connected with the cantilever and the tip. So what happens as we move? This is the platform. So as we move the platform and our sample is on the platform, this green thing is the sample. As we move the platform, so you can see here the platform is not even, right? So there are edges over here, bulges and edges. So as we move this platform, say suppose towards this side, this cantilever will also move. The tip of the cantilever will move, and it will move like this, okay? And you can see that it can give a 3D, so that movement of the tip will be detected by the laser and the cantilever. Okay, as it moves, so a 3D kind of graph will be created, and you will get a 3D image of the sample which is placed on the platform. As the platform moves, the tip of the cantilever will move, and you will get a 3D. I'll show you the image also. So this is how. So see here, if we take a cellular surface, you know cell has many proteins which are cell surface proteins or membrane embedded protein. So the tip, if it moves, it will trace the surface like this. You can see like this, like this. Even here in a dynamic mode also there will be up and down, and lastly you can see this type of images like here F, G. These are the same images of higher resolution. This is 10 nanometer scale and this is 100 nanometer scale. You can obviously see it better in a 100 nanometer scale, the 3D kind of surface structure of the membrane. So the atomic force microscope will give a 3D image. Next we come to the confocal system. So confocal system, how it is different from the normal epifluorescence microscope or fluorescence microscope is that it will before, like this is the normal microscope. So here you see if the surface is not regular, the surface of the sample is not regular, it will get splitted, light will get splitted, and you will see that it is not focused. This is the main, this should be the main focus, but because there are some edges and all, so light gets splitted into different direction and you'll get a blur image because all light rays are not focused on a single point. But uh, to overcome this, we have developed a confocal system where we use a confocal pinhole, so that the detector can only detect the this light source and not the other light sources that arise out of the sample or the lens. So we get a focused image which helps us know the 3D structure. So this, if you see, these are the type of images you can see from the confocal lens, and you can see that it is a 3D kind of image. Next, what was there is the differential interference contrast microscopy and phase contrast microscopy. So here, so here these are the two kinds of microscope. Last two. So in differential interference contrast microscope again, we have a polarizer. Light passes from non-polarized light. Okay, first I'll explain the phase contrast microscope. So there is a condenser and the annulus, and you this is where you keep your specimen. This is the objective, diffracted light, and there is a face plate, and you see with the eye. And the images which you see are of this type, this lower panel. Okay, these are the kind of images you can see in the phase contrast microscope. And if you look at the differential interference contrast microscopy, here non-polarized light will be converted to polarized light, and then it will pass through a set of different kinds of lenses. That is the objective prism lens, condenser, then specimen. We have again another objective and nose piece prism and analyzer. So it will pass through a series of different kinds of lenses, and then you can then it goes to the analyzer. And the type of images is given in the um, first panel. You see, these are the same sample. One is taken in phase contrast and one is taken in DIC, or differential interference contrast microscopy. So the answer is, in DIC you can see a 3D image, because well, this only gives a phase contrast. Well, always phase contrast is like a normal compound microscope which you used to see like in normal labs. Okay. DIC uses these various kinds of lasers and bonders, these ones, to give a 3D image. DIC. This upper panel is from DIC, and it uses different kinds of lasers and everything to give a 3D image. Phase contrast is a normal microscope. It will always give a 2D image. Confocal will give a 3D image, and atomic force microscopy will always give a 3D. So among all these, we have here atomic force 3D, confocal 3D, differential interference 3D. Only phase contrast microscope gives a 2D. So answer is D.